So Dr. Chang received his bachelor's degree from Harvard University, and then he did his PhD at MIT, and then MD at Harvard Medical School. And then he went to Stanford University for clinical training and also postdoctoral training. And then he became a faculty at Stanford University from assistant professor until now he's a professor at the Department of Dermatology at Stanford University. And I think Dr. Chang is not a stranger here. He's familiar to many people in the audience. And he's very young. And as VP Liu mentioned this morning, he's probably the academician elected at the youngest age, at least in biology division. And his research has been focusing on the genome-wide study of gene regulation and epigenetic regulation of gene expression. And he has made several important discoveries on how long non-coding RNA is involved in gene regulation. And he has also developed various technology for this type of study. So let's see his title. I remember he has a very interesting title. And let's welcome Dr. Chang for his talk.
Thank you, Director Chang, for that kind introduction. It's my honor and pleasure to be here today. I want to talk to you about a fascinating topic, and that is the topic of epigenetics. The word epigenetics implies something above genetics, and it is indeed a topic that has captured the popular imagination. Time magazine put this topic on its cover. And this is an idea about the living genome, because all the cells in your body have the same genes, yet your muscle cell is not the same as your skin cell or your heart cell. All the cells are actually making choices about which genes to turn on and off. And that is the mechanism, the function of epigenetics. This field has advanced rapidly, so now we have ways of directly measuring this activity. It is very important because it reflects the dynamics between nature, what you're born with, and nurture, your environment. And as I'll show you, this also has direct impact on clinical states and health effects. Another analogy for epigenetics may be the information transfer when we project a picture through a lens. So you can imagine that the DNA template is this original picture, and this lens through which this information is projected is the mechanism of epigenetics. In a youthful or healthy state, we have this proper transmission of the information, but this can be corrupted with aging or disease. Both the information content in the film or the lens can become cloudy, leading to a corruption of information. But the idea is that even if we cannot fix the genome, we can basically correct the epigenome, or almost like the glasses I'm wearing, to correct the information so that we can restore the projection of this image and the phenotype. This is a very rapidly advancing area of investigation. In different fields of research, we go through phases of discovery, detection, and decoding. The initial concept of a mechanism, a way to measure it, and finally a full scale up of rapid application. This happened in the field of genetics or genomics with the hardware of DNA, discovery of, let's say, the double helix by Watson and Crick, their structure, the first method to sequence DNA back in the 70s, and over the last decade, very rapid scaling up of rapidly sequencing many, many genomes. A similar revolution has happened in the field of epigenomics. The first chemical marks associated with epigenetics were recognized in the 1950s. A first biochemical method detected came out about in the 1980s. And I'll tell you about a technology developed just a few years ago that rapidly scaled up the speed of decoding this information. What we're talking about here is the control of gene expression. When we think about genetics, we think a lot about disease genes. Often protein coding genes are associated with disease. But what we know is that each of these genes are hooked up to many, many switches. These are the binding sites of transacting proteins, transcription factors, and or regulatory RNAs. And these are the gene switches that decide when genes turn on and off, and where and where. In fact, the human genome looks more like this. Just 2% of the information is protein coding, and the vast majority of this real estate is this non-coding space. We also know that the vast majority of the disease-associated variants, genetic variants, actually reside in this non-coding space. How do we find the missing information? And so in every human cell, 2 meters of DNA is packed into a 10-micron nucleus. So if you stretch it out, that's even taller than I am, in just one cell. So there's a lot of compaction. Most of the DNA is packed away, not accessible, except for the active DNA elements that are actually accessed by the machinery. So therefore, a few years ago, my colleague Will Greenleaf and I invented a technology called assay of transposase accessible chromatin, or ATAC-seq. This is used as an enzyme, a transposase, that copies and pastes DNA. And we load up this enzyme with the adapter used for our sequencing machine. We discovered that when this transposase sees chromatin from our cells, it can only copy and paste into the open chromatin sites. Therefore, in a single step, you have selectively and covalently attached these adapters to the active sites, allowing you to sequence directly. This technology led to an amazing, 1,000,000-fold improvement in the sensitivity and 100-fold improvement in the speed of finding active DNA regulatory elements, including from clinical samples. Let me show you some data. Here on the x-axis are genomic tracks. These are locations of different genes. And the y-axis, the height, indicates the level of accessibility, or is a proxy for activity. This is the prior gold standard with the enzyme called DNase 1. It needed 10 million cells. These are the peaks. These are the active elements. It's the first version of ATAC-seq, needed 50,000 cells. And this is the ultimate resolution of ATAC-seq, single-cell ATAC-seq, which we accomplished several years ago. And this is several hundred single cells. I hope you can see that the pattern looks exactly the same in all these experiments. But now we can zoom in, and now each row is a single cell. There's 254 single cells here. At every position, we see either 0, 1, or 2 reads because it's a diploid genome. And so therefore, this kind of analog signal has turned into digital information. When we see these peaks, that says that there's something acting on the DNA and making it accessible. So you might want to know, what is the identity of the factor that is accessing DNA? There's a very interesting quality of the ATAC-seq data. When we zoom in to one of those peaks, we see that at the summit of every peak, there's approximately 8 to 10 bases of a notch. And this is actually the binding site of the DNA binding factor. So if you think about this method as spray painting the genome, you can imagine that if I spray paint a building, but I put my hand in front, when I move my hand away, there's a shadow. It says there's an object there blocking the way. And that is what is exactly happening when a transcription factor sits on top of the DNA. So when you zoom in, for example, to this factor, this is the binding sequence of this factor called CTCF. This is every instance of that site in the genome. You see, these sites have this footprint pattern, and these do not. And this tells us that this is actually bound, and these are not bound, and which is confirmed by a different kind of experiment called chromatin immunoprecipitation. So because we already know the binding motifs of hundreds of transcription factors, we can read them out all at the same time using this technology in an allele-specific fashion. Now, armed with this technology, we want to learn the most we can from a map, the global map of gene regulation, what we call a personal regulome. And here, we're inspired by electronic maps, digital maps of the real world, like the ones you use on your phone when you navigate the world. So we represent the world in different layers. Of course, we have different kinds of land patterns. We know what the buildings are. We know what the streets are, maybe where your friends are. And each of these layers makes this map more and more useful. So inspired by this map, we also want to learn from a snapshot of the cells, what kind of cells are present, the cell states, the tissue microenvironment, the lineage, the drug response, and connect that all together by computation. And so we can infer all of that from this personal regulome. Let me show you some examples of these kind of applications. And one of the prominent applications is in cancer biology. So this slide makes the simple point that cancer cells are all different. On the left is a print, a very ancient one from Virchow back in 1847. He just showed what he saw in the microscope, that these cells all look different. On the right-hand side is a more modern depiction from review talking about genetic changes, actually epigenetic changes in a tumor. So together with my colleague Ravi Majeti at Stanford, we applied this very sensitive ATAC-seq technology to cells identified directly from leukemia patients at Stanford. In this acute myeloid leukemia, we can actually isolate the original hematopoietic stem cell, the cell that has harbored the first mutation, which is not yet enough to cause a transplantable disease. This is the leukemic stem cell and also the blast cell, and also from the same patient sample. We can see from the ATAC-seq maps that actually each of these cell states has a different epigenomic pattern. And moreover, we discovered that this has a very strong prognostic impact. The more this pre-leukemic stem cell resembles the hematopoietic stem cell, it's able to more take over the population and create a worse prognosis. This technology we found also has a very profound impact on understanding disease treatment. This is a disease that I'm showing you called cutaneous T-cell lymphoma, or CTCL. This is the disease that I sometimes diagnose and treat. Here's a patient with this disease. This is caused by a cancer of the skin-resident T-cells. You can see this patient is not doing very well. This is an important kind of application for epigenetic therapy because it is the first approved FDA indication for a class of epigenetic drugs called histone deacetylase inhibitors, or HDAC-I therapy. Despite the fact that this drug's been approved for more than 15 years, it's very much a black box when we treat patients with this particular drug. Only approximately 20% of patients benefit, and it's actually very frustrating because we don't really know why. We think that part of the challenge was that even though this drug is supposed to target chromatin, in clinical practice, we have no way of seeing what is happening to the chromatin at all. So if you imagine that I'd say, we're going to treat hypertension, but we're never going to measure the blood pressure. We'll give some medications, hope something good will happen. You would think that is a ridiculous proposition. But that is, in fact, what is happening in cancer treatment. And so using this technology, we conducted the first clinical study in real time of using epigenomic technology to measure chromatin accessibility during patient response to HDAC treatment at Stanford. And what we discovered is that there's a very simple relationship between clinical response and the chromatin. In the patients that respond, their chromatin immediately gains accessibility, as you would expect from the HDAC inhibitor, and they show a corresponding rapid decrease in their leukemia cell count. If there's an absence of this response, then there's also no clinical response. So this is the first sort of molecularly demonstrated sort of, I think, efficacy endpoint of HDAC inhibitors in patients. Spurred by this example, we were inspired to actually further push our technology. I explained to you how we can measure chromatin changes in individual cells with single-cell ATAC-seq. And what we did next was to combine single-cell RNA sequencing with single-cell ATAC, measuring both RNA and DNA from the same single cell, accomplished using this microfluidic strategy. So this may sound very simple. These are two things we already know how to do in molecular biology. But really, we're trying to do that from the same cell. Okay, so that's kind of like asking you to make a Japanese dish and an Italian dish in the same pot. Make both of them taste exactly the way they should, okay, but one pot, because we only have one cell. And so that's what we had to accomplish with a bunch of DNA gymnastics. So let me just show you that this actually worked. And the technology we chose, the application we chose was actually to sequence the T-cell receptor genes on the RNA level and measuring the chromatin at the same time. And there's a very good reason for that. So as you may know, the T-cell receptor genes undergo somatic DNA recombination. So there are more than 10 to the 15 possibilities in the body. They're the perfect lineage trace for cancer immunology studies, and it's also very important for antigen discovery, for having both the alpha and the beta chain from the same cell, and it's a way to either weaponize or disarm immune cells. And so this was actually accomplished. So let me show you what the results look like. So here, every column is a single cell. Every row is basically a pattern showing activation or deactivation of the transcription factor pathways. These are T cells, and in these cells, we can actually then see T cell receptor alpha and beta sequence with a joint probability of success in 85%. And the sequencing information immediately gives us clues as to basically the classes of T cell receptor that they are in, and therefore possible antigens that they may recognize. And this is a study done in collaboration with my colleague at Stanford, Professor Mark Davis. Okay, who's an expert in T cell receptor biology. So we can now imagine the following scenario, that when we use this technology to look at, for example, patients with cutaneous T cell lymphoma, we can then, from the blood, identify the leukemic clone. They all share the exact same T cell receptor sequence, and then at the same time identify the non-leukemic clone, for example, the patient's immune response fighting back. Okay? And we can then separately understand the gene regulatory landscapes in these two separate populations rather than averaging them together. And this is indeed what is now possible. So in this one of our first cases, this is a patient, again at Stanford, almost a 69-year-old patient with an advanced CTCL, but could not be actually typed by our clinical lab flow for the TCR sequence. So here, what I'm showing you is that every row is a different V beta segment, and every column is every possible VJ segment. But you can see that there's a dominant clone that corresponds to 70% of the clones in the blood, and that is the leukemic clone. So this is the leukemic TCR. And from that clone, we can identify signs that this is actually a memory phenotype based on the certain transcription factors. It's a Th2 bias, and there are other interesting features. We can also see the patients, the immune response fighting back, and evidence of immunosuppression, such as this TGF-beta pathway activation from SMAD3. So this kind of technology, we believe, will lead to a actually very accurate and precise diagnostic for both cancer and we think a lot of other immune-related diseases. It is actually very important to have this clonal specific separation. If we average all these clones together and do the analysis, we can no longer see these clonotype specific changes that are, of course, critical to this interpretation. Okay, so a further and important insight into the landscape of regulation is that gene control occurs over long distances. And so because these gene switches and protein coding genes are interweaved and packed next to each other, it's recently recognized that many of these elements, gene switches, jump over multiple genes to control their target gene. And this case also occurs for inherited single nucleotide polymorphisms such as SNPs. And so finding the true target gene of disease-associated elements really, I think, is a very important problem, and which I call the last mile of human genetics. What is the true target gene of this SNP? In the past, oftentimes, people report, by default, the nearest gene on the linear genome, even though we're not sure that's really the true target gene. Okay? And so the technology we develop expands on chromosome conformation capture. This is a class of biochemical methods that allows you to capture, by spatial proximity, DNA that's next to each other in space. We can capture those, ligate them, and then isolate only those associated with enhancers, which are the active gene switches, marked by this particular histone modification. So the concept is that we can now see this kind of map that this is the causal variant, but it's actually not affecting the nearest gene, but actually maybe these two genes over to the left and to the right. We made a number of improvements that made this technology, again, about 10,000-fold more sensitive and 10-fold improvement on the sequencing depth, so we can get down to 1 kilobase resolution. So we can now, again, isolate from human blood samples a standard blood draw, a small number of cells, and in this kind of a map, it basically is, you want to, this is the genome in one direction and genome again, and so these squares, these diagonals indicate long-range contacts in DNA. You can see that in chromosome scale resolution, zoom in, down to individual gene and promoter level, down to the 1 kb resolution. This is a rather rare cell type from blood, which is possible to have a map like this from a standard blood draw. The traditional technology will require 4 liters of blood from a person to make such a map. For the basic science in the group, adult human has 5 liters of blood. Okay, so you would have to bleed the person dry to actually do something like this. Shouldn't do it. Okay. So let me show you what you can learn from this. So this kind of a map is called a virtual 4C map. This dotted line is the viewpoint from which we're looking at. And then each of these peaks indicates an active enhancer that is in contact with this initial viewpoint. I'm showing you the map, enhancer contact maps in 3 T cell types, naive T cells, Th17, which is an inflammatory T cell, and T regulatory cells, which are somewhat immunosuppressive. These are the Genome-wide Association nominated SNPs for rheumatoid arthritis and type 1 diabetes. It's closest to this gene, SMIM20, but we found that true targets actually in Th17 cells are actually these 2 other genes. This is a transcription factor in the Notch pathway, very important for T cell activity, and this is a calcium channel, again, active only in T cells. So these are actually the true targets. We can, in fact, verify that by using new CRISPR genome editing technologies, actually epigenome editing. We can target this dead Cas9 with a silencer domain to silence either the promoter or this enhancer right here, and you can see both of these strongly reduced expression of this RPPJ gene, indicating that this is in fact a true enhancer for this distantly located gene. We extended this kind of study systematically, and we discovered that the target genes of disease-associated elements are often much greater than was previously thought, based on the nearest gene. Of course, in multiple diseases and overall in autoimmune diseases, a 4-fold expansion of the true target genes. We are taking a number of methods to check these functions in vivo, one of which includes knocking out the enhancer. Okay, let's just leave it down there. It's okay. Yeah, I like that for effect, right? Knock out the enhancer and the thing just falls down. It's very dramatic. Okay. So this is an enhancer that we found to be active only in a particular subtype of T cells. And so we basically, it's homologous in the mouse, and so we deleted this. This is an experiment done in collaboration with colleagues at UCSF and also UC Berkeley. And the result is that this was predicted by our map to connect from the intron back to the promoter of this gene. And in this knockout mouse, this enhancer deletion mouse, we discovered that if we activate these T cells in vivo with anti-CD3 stimulation, there was a defect, a selective deficit, only in T effector cells for upregulating the protein IL-2RA, but not in T regulatory cells. And so this is an important point to emphasize. And that is that this is not the same as knocking out the protein, because you would lose it everywhere. But by manipulating just the enhancers, you can get a cell type specific loss, but while preserving the function in another cell type. And the cause of autoimmunity is actually from the misbalance between effector cells and T regulatory cells for this activity. This is a mouse example, but I believe that the most powerful path going forward in the future will be directly to study the patients. This is a study using a patient's material, but from, in fact, a GWAS study. This is a cardiac smooth muscle cell from a patient who is a heterozygous carrier for a DNA variant that's associated with heart disease. And what we discovered is that these 2 alleles, you can actually identify the enhancer connection map in the same nucleus. The G allele, the risk allele, indicates a connection to this gene SMAD3, whereas the alternative allele, A allele, does not. And so because they're in the same nucleus, they act as perfect controls for each other, indicating that that point mutation acts in cis to change the gene regulation pattern. What I've told you so far then is that these new technologies enable a personal global positioning system for navigating the gene regulation landscape, the Regulome. Our concept can directly, very quickly go from a patient sample to these novel technologies to map out the switches that impact disease. And that could give you potentially a custom therapeutic strategy. The most exciting aspect of epigenetics is not only can we learn about what is happening now in the real time, but also possibly then to change it with something we might do. So what I'll tell you next is something I'm excited about, a new class of regulators in the epigenetic space called circular RNAs. Circular RNAs are abundant and ubiquitous, and it's a newly recognized class of non-coding RNAs. If you imagine that exons 1, 2, and 3 are splicing forward, if exon 3 goes backwards, this will create a circle. Okay? And so they arise from back splicing, and we now know that the human genome has over 10,000 different circular RNAs. They are incredibly stable because RNAs are degraded by ends being chewed up by exonucleases. Circular RNAs, by definition, have no ends, and therefore they can last for days instead of for hours as a traditional linear RNA. There's some evidence that they can function as sponges for RNA binding proteins or microRNAs, and also templates for translation. But it's also equally important to recognize that in addition to these endogenous human circular RNAs, there are actually viral pathogens that have circular RNA genomes, such as hepatitis D virus, plant viroids, and also other pathogen genomes. So this suggests a fundamental problem. How do cells distinguish self versus foreign circular RNAs? We stumbled upon this problem first by actually thinking that we could use circular RNAs as a gene expression platform. And so a very talented postdoc in the lab, Grace Chen, created a way to make circular RNAs in the test tube. This uses the cell splicing intron of the bacteriophage T4. By permuting the order, we can actually make this thing splice itself backwards, and the internal ribosome entry site is placed up downstream of GFP, so only when this becomes a circle can you have a possibility of reconstituting the translational template like this. That just shows you that we actually made this circle in a test tube. RNase R is a very processive exonuclease. It will destroy all linear RNAs, but the circular RNA is basically impervious. That's the evidence you have a circle. We can sequence the junction and show that we have precise recapitulation of the ends. Okay, so we can make a very beautiful circular RNA, but when we put this into cells, we could not get GFP expression. Instead, what we found was that there's a massive immune response. Okay, so this is left, I'm showing you data from keratinocytes, right from macrophages. The linear RNA is the exact same sequence, but with a point mutation in the intron, so it can't splice. Okay, you can see that if we use the circular RNA of the same sequence, we get like 200-fold activation of a number of immune response genes, and the macrophage is like 1,000-fold difference. Strong induction of the genes. And this is not a property of the stability. This is the way it is even after the first 24 hours after you introduce these RNAs. So circular RNAs was present, we found in nearly every cell type we tested, and that this turns a full suite of antiviral and inflammatory genes. This has some potentially, we immediately went on to ask, is this sufficient to actually confer a function? So this next experiment is to ask, what would happen if we inoculate cells with circular RNAs while we try to then give them a viral infection? So this is using a reporter virus. This VEV is an RNA virus with a GFP, and if it gets infected, the cells will turn green. The RNAs are now labeled with a different color, Cy3, and this is a flow cytometry experiment. So you can see that we can put in these RNAs, linear RNA, circular RNA, same sequence, right, as before. But you can see that if you put in a linear RNA, no protection against infection, circular RNA, nearly complete protection against infection, both in the cells that took up the circular RNA and also in bystander cells that were just in the same dish. Okay, and that is shown there. And as results quantified here, a 10-fold reduction in overall infection, and even in negative cells that are just bystanders that are protected by viral infection from the circular RNA immunity. So this ability to activate a powerful antiviral immune response is very reminiscent of also the way that we can actually get adjuvant activity for vaccines. We heard a lot about vaccines this morning. Oftentimes, when you want to induce a antigen-specific immune response, you have to add a non-specific adjuvant. And so here, this is a model experiment using circular RNAs that we made in the test tube, but inject mice with chick ovoalbumin as a model antigen to then attract antigen-specific T-cell response and antibody titer. So you can see that here we use, this is a T-cell or antigen-specific T-cell response, CD8 positive, interferon gamma positive cells. This is the negative control, positive control with poly-IC. You can see a potent induction with circular RNAs, and this is actually a different flavor packaging the RNA. We also see this very potent induction of antibodies against ova induced by the circular RNA adjuvant. Okay, so these experiments hopefully convince you that circular RNA immunity is functional and actually can be harnessed as a way to manipulate the immune system. And so now it comes back to the question of how. How does this happen? When we think about self versus foreign RNAs, we should really think about broadly two classes of responses against nucleic acids. One class are sequence-specific mechanisms, such as RNA interference or CRISPR, which detect specific sequences and then fight back. There's a second class of RNA immunity that's based on some sort of general structural property, which is shown on the slide are, for example, let's say a sensor called MDA5, which senses long double-stranded RNAs, or this other sensor called Rigi that senses 5' triphosphate in viruses. They're non-sequence specific, but they sense specific structural properties of RNAs. This is a review from a colleague and collaborator, Sun He, at Harvard Medical School. And so we went on and asked what are the basically features. I can tell you that it's not through RNA or CRISPR, but it's one of these structure detecting mechanisms. We're very fortunate in that I told you that we can detect circular RNA immunity in almost every cell type we tested, except this one cell type, 293 T cells, which appear to lack this response. Therefore, we put in different sensors into these cells, and we found that Rigi, when it's added back into these cells, potently recovered circular RNA immunity, but not MDA5 because he's completely dead. Conversely, we went on and knocked out, we actually used the Rigi knockout mouse embryonic fibroblasts from the Kira lab. And you can see this nearly completely abrogates the immune response to circular RNAs. In all these genes that we tested, this second row is the knockout, and that's here. This is like a 6,000-fold difference. It's completely dead. We also knocked out the human Rigi in HeLa cells and show, again, the same result, that Rigi was necessary for circular RNA immunity. In fact, we can show that Rigi and circular RNAs become intimately associated and form these foci only with circular RNA, but not with linear RNA, and this is quantified here. So the Rigi is functionally required and also very proximal, we believe, to the sensing mechanism of the circular RNA immunity. The fundamental question in any kind of immune system is what is the basis between foreign versus self-circular RNA? And so that is what makes the foreign ones different from the ones that our own body is making. Remember, I told you at the beginning, our own body makes 10,000 different kinds of circular RNAs. So first we discover that the circular RNA immunity is sequence independent. In the beginning, I was showing these experiments with green fluorescent protein hooked up to an iris. And so if we swap that out with M-cherry, we can also make a circular RNA, put it into cells, equal activation. And so basically it's completely independent of any sequence. Any sequence that we make in this way into a circle will activate immunity. We can further show that other features important for other known ligands, like 5' triphosphate, double-strand RNA structure is not involved, and that the circular topology is required for immunity. If we make a circle and then cut it with RNase H, put it into cells, it'd be completely dead. So it's not any other product coming along in the reaction. It has to be the circle. So So this is drawing kind of the reaction. These are the introns that programs the circle that gets made. So if the exon, that's the circle, is not relevant, that by logical elimination, it must be the introns. And that's in fact what happens. We found that if we use a human intron to make a foreign circle sequence, the exact same sequence, now we completely change the immune profile. So the circular RNA GFP will actually induce basically a 1,000-fold induction of these immune response genes. But if we make the same circle with a human intron, then no reaction whatsoever. Okay, so the intron identity somehow confers or regulates circular RNA immunity. So the next question, which I think is the key experiment, is that what happens if you make a circular RNA with both a foreign intron and a human intron? Which one's dominant? And the answer is that the human intron would dominantly confer circular RNA identity, self-identity in cis. So it will win out. So anything that has a human intron, foreign sequence, foreign intron, now there's no immune response again. And so and we further, in other experiments, show that if we put these two kinds of circles, but the introns separate together, the immunity will start activate. So it has to be on the same molecule. Okay? And so as I've shown you, since this is the intron, but this is the circle, and this is the part that's going into cells, there must be something left onto the circle that transmit the memory of its biogenesis. And so in the next experiment, Gray showed that we can pull down the exon circle and perform mass spectrometry, a technology we developed called CHIRP-MS. And we can discover there's a whole set of proteins that are associated with endogenous human circular RNAs. If we make a foreign circle, but with a human intron, we also basically tag those circles with these self-proteins. So the splicing will load proteins onto self-circular RNAs, which is highlighted here. So in summary then, what I told you is that the mechanism of circular RNA immunity is a system based on RNA biogenesis. In fact, specifically monitors the introns that program the circular RNA. If it's a foreign intron, this will create a naked circular RNA, it will activate Regi, and create innate immunity. If it's an endogenous intron, it will actually tag with the circular RNA with proteins and other chemical modification, which will block Regi and then prevent this reaction. And therefore, the intron, which is really the largest part of the transcribed non-coding genome, in fact, is the arbiter of self-non-self identity in this RNA world. Summary, this is what I told you. Circular RNAs are now a new ring of power. I hope some of you recognize what this is, and because it represents a new form of biology in circular RNA immunity, and also a new mechanism of manipulating the immune system, perhaps for therapy and for diagnosis. I'd like to thank many colleagues, talented colleagues involved in this work, and acknowledge my collaborators and funding sources. Thank you for your attention.